Looks like it is. Hey, Alice.
How are you?
As you are on a Monday morning.
Okay, it's not working very well. Okay.
Cool. Good to see you all again. Welcome back to Neuroccino on the 1st of September 2025, the first edition of the new academic year. And the rules of the game, just to recap, everyone is welcome to join us and present a paper, a book chapter, some piece of science they get excited about. And then we discuss it to the best of our knowledge within half an hour. And today, Lilit is going to kick us off with cortical reorganization after upper limb amputation. Lilit, take it away. If you can also share the link in the chat for everyone.
Okay,
that'd be-
here is the paper, and I'm going to share the screen. Let me know if you can see the screen. Okay. So hello everyone, and this is the first academic year Neuroccino, and I'm excited to share you this paper that was published just over a week ago in Nature Neuroscience by Hunter Schoon. Some of the authors maybe you're familiar with, but especially this one, because a couple of Neurocinos ago I presented you a paper with the title Against Cortical Reorganization. It was very rich review with debunking of a lot of textbook classical examples of cortical reorganization. It's really nice paper. I really suggest you to read it. And in that paper, they also talk about in more detail what do we actually mean by cortical reorganization, which is essentially when a cortical area starts to respond to novel input that was not responding before. And in that paper, they basically went step by step and logically showed that this is not the case in all the examples that they demonstrated. This paper is in the same context, but now they are debunking another classical example with their own very unique data set. What is this classical example? Well, maybe you know about it. So, for example, when a hand is amputated, okay, for example, this part of the brain that is responding for the hand sensations and movements, if it is a motor area, then the face region that is lateral to that region is expanding, and now this region starts to respond now also for the face, okay? And therefore, authors before this paper were claiming that this is what we have. This is the cortical organization. We get it, okay? And this paper is aimed to show that this is not the case. By the way, this homunculus representation is, I think, outdated. I don't know what happened with that discussion later, but again, a couple of Neurocinos ago, I presented another paper that was showing the updated homunculus where things are constructed or represented not in a linear way, but in a concentric circles. And also there are inter-effector regions it's like a network that was activating simultaneously, even when you were not stimulating that region directly. And that was, again, still in that S1 or M1 region. So just advise you also to look at that paper. But anyway, this is the example. And what they did they recruited longitudinally. This was surprising for me that this hasn't been done before. We never had any data longitudinally showing that there is cortical reorganization. This was always cross-sectional studies, at least in humans. And in this case, this is the novelty. One of the novelty of this study is that they recruited 3 participants, okay, and they scanned them at different time points. For the first patient, for all the patients, they scanned 2 times, twice before amputation surgery. For patient 1, they scanned it after 3 months, 6 months, and I think 1.5 years after amputation. For the second one, 3 months, 6 months, and 5 years. For the third one, it was only after 3 and 6 months. They also recruited healthy controls, so they had their hands were healthy, and they recruited them at 3 and 6 months after amputation surgery. So they did that, and what they asked for the patients, that you can see them here, they asked them to move both intact hand and phantom hand. Move, not imagine, but move them. So non-existing hand. And this is another surprising fact was for me that actually there is a difference between imagining of moving non-existing hand and moving existing hand, non-existing hand, okay? And here you are seeing a contrast between moving the phantom hand versus imagining moving the hand. So you see 3 patients. By the way, the second patient, you will always see the left hemisphere because for that patient, they amputated the right hand. Okay, and for the first and third, it was the left hand amputated. So yeah, you see that there is a difference. So fact number 1, moving imaginary hand and moving non-existing hand and imagining non-existing hand is different in terms of at least activation. So then they ask a question, okay, we amputated the hands, and is there the difference? Something happened, maybe the activation becomes less or it disappears. So what you are seeing is the contrast between hand and rest. No, hand against feet movement and lip against feet movement. Before for 3 patients and after amputations for 3 patients. And already you can see kind of qualitatively that it seems there is not much of a difference. Okay, so the activations stays pretty stable, both for lip and hand. But they went a bit more in detail into this. And what you are seeing here is the following. On y-axis, let me move it a bit closer. On y-axis, you see the activity amplitude. On x-axis, you see different segments of supplementary somatosensory area. So this strip over here. More medial over here, you have like a feet area. More lateral is like face and lip. So basically, this region should respond for lip movements somewhere here. And this region is the hand. So they did it for the full hand and for all the fingers separately. The lines represent different sessions. So the black ones is the 2 sessions before surgery, and the rest is at different time points. And as you can see, it seems that there is no difference again in terms of the activation strength of the hand. And this is true for all the patients that they had. next piece of the figure is this one. What they did, they calculated so-called COG, which is center of gravity metric, which is basically, as I understand, is a metric that will tell you if there is a shift in space of the activation. Okay, so for example, if you have positive values, then the center of gravity, like the main activation, moves towards the feet region. If it is going lower, then the shift of activation goes to more to the lip area. The gray violin plots are representing distribution of healthy controls. So, and the different signs here are just the 3 patients. So you see that also in terms of shifting the activation, like especially they are still within the normal range. Okay. Then they went and they tried something more fancy, let's say. They trained a support vector machine classifier. How? So they took the sessions before amputation. They trained the classifier to distinguish between activation of different pairs of fingers. So for example, you have finger 1 and 2, and they trained the classifier to tell if a classifier can classify if this activation belongs to finger 1 or to finger 2 on data that before amputation. When they trained it, then they test it on data after amputation. So they gave again some data set a bit of finger 1 and 2 of activation, but already after amputation of phantom hand. And they asked the classifier to classify the which activation belongs to which finger. And it was pretty high, the accuracy. Maybe in these cases it was pretty low. This is the classification of finger 4 and 5. I don't know how are your fingers, but when I push my pinky, all the rest fingers goes down. So maybe because of that. So if you try to do like I have this connection of 2 fingers. And probably this is what's happening here, and the most easy distinguishable are like these 2 fingers. For the patient 2, I guess, yeah, for the patient 2, it was a bit more lower the accuracy of the classification. But yeah, they say that selectivity still preserves even after amputation in terms of cortical activity. Again, this is a movement of the phantom hands that let me see what else I can show. Well, they did all analysis in the same style. But for example, for the lip, you see that here now they ask to move the lips, and you have the activation in the lip, and there is also the same level of activation across the sessions. Again, compared to normal control groups, it was also in a range of normal, in the normal range, the variations. Then here they show the boundaries of the lip for patient 2 and 3, 1 at different time points. And you see that the boundary is seems to stay the same. The different colors represent different sessions. And then they also like measure different distance, thumb to lip, also within the normal range. Then to validate, they took chronic amputees group from other studies, and they calculated the COG again, and you see that it is within normal, the center of gravity. Both, this is the normal subjects, the healthy subjects, and this is the subject with the chronic amputees, and the 3 dots are the patients of the current paper. And everything basically the same under the same range. So their conclusion, it says that our longitudinal approach reveals no sign of topographic reorganization in S1, not even subtle upregulation from homeostasis, further reinforcing the notion that S1 is not governed by deprivation-driven plasticity. So basically, this is a great news for BCI and like companies like Neuralink, because it means that those regions are not reorganizing and they stay the same. We just need the stimulation. And for example, in general, prosthetics also, it's great news for that. This paper has a lot of supplementary materials and extended data. First, I want to show you that they also did this for motor area. And again, it was all the same, like nothing is changing across sessions. They did the same for non-amputated hands and also finger variability. Let me see. Yeah, and the last thing maybe to show what was also interesting, that this is subjective feeling of fingers. And you have here the feeling of pre-amputated hand. And then you see that the feeling of phantom hand is stronger than pre-amputated hand. And also in terms of pain, also, for example, for patient 2, the pain becomes the maximum, as I understand this plot, which is also interesting. So the activation stays the same, but the feelings and the pain increases, I think, as I understand. So yeah, let me let me know if you have any questions. But yeah, this is the paper, was very pretty short, but informative, interesting, and yeah, happy to answer your questions and discuss this.
Thanks, Lilit. Very impressive work, especially given that it's only three patients. It's extremely deeply phenotyped. Hands are flying up. I don't know who was first, but let's go with ladies first. So Anna.
That's the lady, Mikael, next to me.
Mich then.
Sorry. How do they, it's very interesting study, and thank you, Lilit, but how do they demonstrate formally that there is no difference in the activation? Because as far as I see, you don't see any difference, but that might totally be due to a lack of sensitivity of the methods and doesn't seem to be controlled for.
Well, they did some tests. I am not familiar much with the Crawford version of t-test, but they had some t-tests. They were uncorrected, which makes sense maybe, but yeah.
Absence of evidence doesn't mean significance of absence.
Yeah.
What I'm trying to say is like, the best way to tackle these kind of questions is to run Bayesian statistics and demonstrate that the absence of difference is not related to the noise.
Yeah, the one that the method more advanced that they used apart from like t-test. Well, they also did correlation, but I think this one was an interesting approach where they trained the model before amputation and they tried to predict to which finger belongs this activation. So the model didn't see the after amputation data, but was still pretty accurate in telling it apart. And they also did the vice versa. So they trained it on post-amputation period, and then they say, okay, now predict the activation of hand that was before. So I think this method was pretty stronger than others. But yeah, I get your point that in a Bayesian way you can say if there is no difference. But I don't know if you can do it with just 3 participants.
That's a good question. But the participant with himself, you can always run the analysis.
Yeah.
And my other question, and this comes like in stark contrast with the work of Cornelius Weiler, if I remember well, who demonstrated these contralateral activations in patients that were non-amputated but had a stroke in their brain that disconnect the motor cortex. Is there a discussion in the paper related to those different mechanisms where you have a peripheral amputation or central nervous system disconnection, and how does this impact the remapping in the brain?
No, there were not, as far as I remember when I read, there is no discussion about disconnections. The only thing that they talk about is the muscle constructions were still there.
Okay.
And they also have videos of the patients asking to move their phantom hand. And you see that there are muscle movements. And also some patients had, some of them, maybe 1 or 2, I don't remember, had some type of surgery where they tried to reconnect the nerve innervation things. Not really familiar with that procedures, but maybe that also can affect some differences. But yeah, there is nothing about disconnection and cortical remapping, reorganization about that. But for example, I looked at these figures. Let me show you one sec. Of unaffected hand, and it seemed like there is more. There are so many plots. Sorry. See. It seemed a bit more choppy. For example, I don't know if this is a thing, but it is unaffected hemisphere. And I don't know, it seemed a bit more choppy, the contralateral, than the one that should be affected. So yeah.
Did you go specify? Okay, thank you very much. And a very last question, why were we missed that part? Why were they amputated?
There were different reasons. So one of them, it's all here.
Agree? Did they agree?
Huh? Yeah, they agree. This is
The
great.
consent form.
Where was the information? Yeah, here, different reasons. Here.
Just a different reasons?
No,
tumor. Okay.
One is tumor, another one something else. So, and yeah, I think it was here, and also this was also maybe important. Some of them use prosthetics, some don't, but yeah, feels like we need more data. But at the moment, even this already tells us some hints.
Unique data set, but if any of you wanna contribute, we can do it in Bordeaux.
That's taking the love for science too far, mate.
Sacrifice we're ready to do for our science.
Right, there is two more hands up, Chris and then Fernando.
Yeah, thank you. A very interesting paper. So a little bit on the question about the stroke. So there are a bunch of there's a lot of literature about plasticity. How, I don't know if they discussed that, but how would you explain that there is no plasticity here, wherein plasticity seems to be explored? Could it be because the mechanism of or like basically disconnection is much further, so the disconnection is from the actual limb, whereas when plasticity is studied usually with stroke, for example, it's a disconnection directly in the brain. So would there be a mechanism because there is like a damage in the brain, so then there would be like an activated plasticity, or would it be more because of another because of maybe the region where it is, like would motor areas not be very plastic and other areas would be more plastic? Or I don't know if they discussed that.
No, they didn't discuss that. This is pretty very brief communication as is written here, but you touched really important difference. So in the paper with Makin and Krakow, the ones that against cortical reorganization, they also like their um, highlight that the difference between plasticity and cortical reorganization is different. Cortical reorganization, they mean that a region starts to respond to totally novel input. So for example, it was responding before only to visual, and now it starts to respond only for auditory, for example, or like it changed the identity. While plasticity, they don't argue against it, as I understand, because plasticity said that it can upregulate, that the circuits were already responding, but they were not the main, their job to respond, for example, to visual. They just get unmasked and they become upregulated, and now they appear as if they changed the identity, but they were already responding to it before. So in terms of, in this case, they maybe were expecting to get some type of upregulation, but they did not get it. They are not discussing it why, but I think I agree with you, probably because when we have peripheral limb, amputated, then it is not directly affecting the brain. So the brain stays the whole and the constant, so nothing is changing. What is changing is like only the peripheral parts of the body. So probably that is why maybe there was no any plasticity mechanism happening. So plasticity exists, but not according to Mackin at the moment. All the evidence that they looked at in that paper and in this paper, it's not actual reorganization, just, yeah.
Thank you. Yeah, I didn't know about the difference between plasticity and reorganization.
Yeah, it's also, I didn't know that, and I don't think about it, but then after reading that paper, I'm like, yeah, makes sense.
Yeah, that's
Kind
interesting.
of. So yeah, plasticity in that sense, but also it's also a question of terminology, but it was nice that they defined it and it makes things clear, at least for me. So
And then
Right.
last question.
Yes.
Also in similar lines, but I feel like the main goal or one of the goals of this paper is to challenge the long-held assumption that there's cortical reorganization. There's a takeover of, how to say, a repurposing of a cortical area for a function. And I think they look at they mostly look at the function of the hand and I was wondering if they do look into any other, like in any function takeover, maybe they could also look into the contralateral hand or movement of other limb and see how if there's any change in the cortex across that.
No, that's a good question, and yeah, they also did all the same analysis for contralateral hand and the unaffected hemisphere, for somatosensory area, for motor area, for the lip, everything, and it's all seems stable and in the same way, nothing is changing. See, like the lines are overlapping heavily, and these are difference periods. And this is also amputated hand and finger, like they did for the same thing that I told you, just for also the other hand. And maybe this plot, I don't know honestly why this is the last page. I think this might be even important. They're trying to explain why it happened. That we think that there is takeover of the lip area of the hand. And their explanation is that when you're not including it's like artificially created this problem. When you're not including hand in the analysis, this winner takes all automatically has to assign to something, and it is assigning to the lip, but the hand area is still there. It's just you didn't add the condition in the analysis, and that is why obviously will be no. But so the first probably problem was that idea that when you're moving the hand, phantom hand, there is an activity and you have to include it in the analysis. Then there will be no lip taking over that region. I hope this is clear. This is the way I understood. And yeah, and also the center of gravity also would shift if you don't include hand condition in the analysis. So yeah.
Yeah, I was mostly wondering, like, if they're kind of criticizing this idea or trying to state something against it. I was wondering how they also look into the part of check if something is actually taking over or not. But it's nice they also have a part like this.
Well, for hand, lip, for hand and lips, they and the green one is fit. They did the analysis and they show that there is no taking over, at least for those regions. So they are specific about that because this was one of the classical examples, and they focus on this. Probably they will. Yeah, maybe they do other experiments, but at the moment they show this, and one of the reasons why not, first, because people were not having longitudinal data, and second, they were excluding hand activity from the analysis. That is why one have to take it over. But yeah. But it's just artificial taking over, not actual. So yeah, that's the story.
Thank you very much.
Thank you so much, everyone. That is all the time we have. Good to see you all again after the summer break, and also excited to share that we just hit the 4,000 subscribers. So clearly this is of interest to some people out there, so very cool. And we will see you again next Monday morning, 9:30. Thanks so much for presenting, Lilit. Have an exciting week in science. Bye.
Bye-bye.